So, good evening, everyone. Welcome to today's week's 7 live tutorial class. So, we'll be starting today's class. Yes, so welcome to week 6 experimental biotechnology live tutorial session. So, hello everyone, I'm Ananya Chatterjee, your teaching assistant for this NPTEL course, which is being taught by Professor Vishal Trivedi from IIT Guwahati. So, this tutorial class goes every week, Tuesday 6 to 8 pm. If you have any doubt regarding those classes, regarding lectures, regarding assignments, if you face any problem during solving the assignment, please feel free to discuss in this class and get your query or doubt cleared. So far, week 1 tutorial has been completed, week 2, week 3, week 4 and week 5. So far we have covered 5 tutorial. If you so all these tutorial sessions or live sessions I upload in a YouTube channel. The link is there. If you have missed or if you want to revisit those videos, feel free to visit the NPTEL YouTube channel, where you will find those tutorial sessions and you can go through. And also in these classes, basically we focus on discussing assignments whichever you have already solved, whichever assignments you are going to be solved, that answer also we discuss in the next year, when the next class, when your deadline is over. But before deadline we can't tell you the answer, but if you are facing any problem regarding understanding the concept, please join this tutorial session, because I also discuss the class material, so that, because every assignments questions are based on class materials only. So if your concept is clear by listening to class materials and understanding those, it will be helpful for you for those assignments, and making your concept clear. So, and also all these tutorial classes presentations are being uploaded in this Google Drive folder, so you'll be able to access all the information from there. So today we'll be discussing week 5 assignment, which is related to gel filtration chromatography, which you have already submitted. But before previous class I discussed various questions, multiple questions from previous year assignments, from this year assignment, and you can see that from those discussions it will be easier for you to solve these questions, because these questions are very much similar to previous year assignment question also. So now the week 7 lecture is live. So today I'll be discussing the concepts of week 7, and also I'll be discussing the assignments of week 7 for previous year, so that it will be helpful. The questions will be related and similar, so it will be helpful for you to solve this year's week 7 assignment which you have live. So the first question is, gel filtration chromatography separates protein on the basis of their charge, size, hydrophobicity, affinity. So you what would be the answer, because we discussed the gel filtration chromatography principle. So in the case of gel filtration chromatography, we have different size of bed volume. So where if your protein size is small, it will pass through this bed volume, and if your protein size is big, what you will do, if you have this kind of porous structure, the bigger protein will not go through this porous structure, it will go through this, it will try to pass through this. So larger protein will come out first, but smaller protein will be traveling more path, so will be coming later. So gel filtration chromatography separates protein on the basis of size. The total volume of a gel is equal to sum of pore volume, void volume, and elution volume. Pore volume, protein injection volume, void volume, protein injection volume, void volume, elution volume, pore volume, void volume, protein injection volume. So the total volume of gel, that means this porous bed where your saphadets and this kind of like beads are there. So those total volume of those bead or gel is obviously not protein injection volume, because protein injection volume is not part of this gel. Protein injection volume, you inject the protein in the column, and it won't be considered in the case of gel volume, because it will pass through a lane or chain, like one column. So that is not that will directly go through this, but protein volume is not a part of this gel volume. Gel volume, the part is the pore volume, where your beads are. So this beads has also pore, so this volume will also be considered void volume and elution volume. Okay. Given below are Kd values of different protein. Protein A, Kd equals to 0.5. Protein B, Kd equals to 0.8. C, 0.3. D, 1. So from the options given below, choose the correct order of elutions of these proteins from gel filtration column. So we know the Kd value is lesser uh, so that will elute first. So here the Kd value is 0.3, so 0.3, then 0.5, then 0.8, and then 0.1. So the answer will be 3, 1, 2, 4. So now what is distribution coefficient? Distribution coefficient Kd in gel filtration chromatography is independent of what? Is independent of void volume, is independent of elution volume, column length, and pore volume. So the distribution coefficient, that means your solvent, like solute, how whatever your protein of interest or molecule of interest is, how it's getting distributed between the solvent phase and the solvent phase and the resting phase, like which is not moving, the mobile phase and in the resting phase. So the answer will be is independent of the column length. If distribution coefficient Kd greater than 1, then we discussed in the previous class that if distribution coefficient Kd is greater than 1, that means protein is adsorbed on the beads and will not elute. So every protein will be adsorbed onto the beads of the gel system, and will not elute. Okay, the answer will be B. Choice of column in gel filtration chromatography depends on charge and mass of protein, pressure limit of the equipment and charge of protein, pressure limit of the equipment, and size of protein. So it depends on the size of protein, and also you have to take care of the pressure limit of the equipment. Okay, because if it doesn't endure much pressure, then that will also not be good, because you are applying buffer, you are applying through wash and all, so it's required to have some, you need to know the pressure limit, and that is how you choose your column. Which matrix is used for protein of 300 Dalton? So only 300 Dalton. So that means the protein bed volume will be able to resolve this less amount of size. So this will be G10, where your protein volume covers like from very less Dalton to like from 10 Dalton to certain Dalton. So there were list in the previous class tutorials where you can find that G10 we can use for very smaller size of protein. Which of the following follows a linear relationship in gel filtration chromatography? So options are elution volume and logarithm of distribution coefficient, logarithm of elution time and distribution coefficient, logarithm of molecular weight and distribution coefficient, elution volume and distribution coefficient. So in the gel filtration chromatography, the linear relationship we have read about is that logarithm of molecular weight of the protein and distribution coefficient. But like what is the size, that logarithm and distribution coefficient, this is the linear relationship. What is used to calculate void volume of freshly packed Cephadic 75 column? So the void volume you can calculate using blue dextran. Which of the following is used for calculating total volume of a gel filtration chromatography column? The total volume you can calculate using acetone. So the void volume can be calculated using blue dextran, and the total volume can be calculated using acetone. Which of the following is done to remove precipitated protein from gel filtration chromatography? Yes. So what you can do, so options are washing column with 20% ethyl. This basically is done to store the gel bed in like it doesn't allow any microorganism to grow. It helps in storing the bed, but it doesn't make sure that you are removing precipitated protein. Washing column with buffer, only buffer also doesn't make sure that your precipitated protein will get away. Flowing small volume of protease through the column. So if you are flowing protease, that means which degrades the protein, so those precipitated protein will get chopped off by protease, so that's why it will help in removing the precipitated protein. Okay, answer will be this. Which of the following experiment setup is suitable for determining the oligomeric status of protein? So what is that? Options are affinity chromatography following native page, gel filtration chromatography followed by native page, gel filtration chromatography followed by SDS page, gel filtration chromatography of denatured protein followed by page. So if you are doing gel filtration chromatography, that means at not denaturing step, at normal stage, so you will be able to know the oligomeric state. And if you run those sample by SDS, that means you were reducing those bonds, you were denaturing the proteins, that means you will get monomeric bands. So from that you will be able to determine the oligomeric status, because if your protein is coming at 240 kDa in the case of gel filtration chromatography, okay, but while you run the gel, it's you are getting band at 40 kDa. That means what is the oligomeric state? 240 by 46. Okay, so this is how you calculate. So it will be gel filtration chromatography followed by SDS page. Next question. A protein from Mycobacterium tuberculosis E37RV is treated with 3 molar urea. What will be the resultant effect on the gel filtration chromatography of urea treated protein X with respect to untreated protein X? Okay, so you have there is a protein called X. Okay, you have applied 3 molar urea. So what do? Urea denatures the protein. Okay, but you need a certain amount of urea to denature the complete protein. Okay, if a protein is conformation is like this, and you are applying 3 molar urea, some part of the protein will get denatured. Okay, so you are seeing that the for the globular protein, the size is getting bigger, so the hydrodynamics value or size is bigger. So what will happen? So what will happen in the case? Options are urea treated protein X will elute after untreated protein X. Urea treated protein X and untreated protein X will elute at the same time. No way. Urea treated protein X will elute before the untreated protein. So this will be the answer, because your urea treated protein size has become larger, so its hydrodynamic value has become larger, so it will pass before the small folded protein. Okay, so the answer will be this. When a protein is subjected to a very high concentration of denaturant, which of the following is true? So what will happen? Then the protein will be totally denatured, so the size will be very large, so then all the protein will come at void volume. A protein was incubated with a suitable ligand, and the resultant sample was analyzed using gel filtration chromatography. The chromatogram resulted in single peak. What can be the correct reason for that? So what happened? You have a protein, okay, you have incubated with a suitable ligand, and the resultant sample was analyzed using gel filtration chromatography, and the chromatogram resulted in a single peak. Which is the correct reason? All the ligands was bound to the protein was denatured, protein precipitated in column, protein was absorbed on the bits. These are baseless, right? This cannot be happening, so then you won't be able to get any peaks. So answer will be all the ligands was bound. So all the ligands was bound with your protein, so protein plus ligand has come out in a single peak. Okay, so today's class will be also discussing previous year week 7 assignments. So for this year week 7 assignment is live, so by discussing previous year questions, it will help you in solving this year's question, but because questions are very much similar and concept is similar, so we will be discussing these questions, but you won't be able to answer until I discuss the lecture's content. Okay, so at first I will discuss week 7's lectures content. So week 7 lectures content is immunology by Professor Vishal Trivedi. I think you have already gone through those lecture slides, but if you have faced any problem, I just would like to revisit those classes once, so that we'll again go back and solve those problems. Okay. So overview of immune system. So immune system basically, what is this? Just a second. Yeah. So overview of immune system, the system which protects the organism against invading pathogens. So we all know about immunity, immune system, because if any we're suffering from any disease, or in everyday situation some microorganism are attacking us or anything happening inside our body, those immune cells are fighting. So okay, so this is the immunity of every organism's health. So there are some specific reactivity induced in a host by an antigenic stimulus. So when we get antigenic stimulus, those immune responses appear. So two kind of immunity: humoral mediated immunity, cell mediated immunity. Antibody complements and other humoral components mediated. So this humoral mediated immunity are basically soluble proteins mediated immunity, which are in solutions, which are antibody complements and other humoral components. Second, provides defense against bacterial pathogen and viruses. Okay. And what is cell mediated immunity? Involves cell such as T and B cell, protection against fungi, virus and facultative intracellular bacterial pathogens, provides immunity against cancer. So what do cell mediated do? Cell mediated immunity do? It involves cells such as it involves cells such as T and B cells. It protects against fungi, virus and facultative intracellular bacterial pathogen, provides immunity against cancer. Now let's understand about antibody. Okay, so from the very like your previous studies, you have been learning that antibody is a Y-shaped molecule. Okay, so here what is this Y-shaped regions? So you can see this blue colored are the heavy chain of antibody. Okay, and this pink color are the light chain. So heavy chains, there are one hinge region between two heavy chains, which are and those are linked by disulfide bond. Okay, you can see those disulfide bond, and this region is an effector region. So in the case of and these are light chains. So in the case of heavy chain and light chain, there are variable change and constant chain, variable domain and constant domain. Okay, and one variable domain and another variable domain of L, this region is called antigen binding site. This is antigen binding site. And you can see if this is the antigen, then antigen with antigen binding site, antigen will get bound to the antigen binding site. Okay, and this is light chain, you have already discussed. So this is heavy chain. So some part of the antibody are glycosylated also. So this is meant by these are glycosylated heavy chain carbohydrates. So this is heavy chain, and these are carbohydrate attached to the heavy chain. Okay, so this is how our antibody we can explain schematically. Now production of antibody. So we'll discuss the 4 steps to generate antibody in organism. At first, lag phase. What is this? When a pathogen or any infectious particle or immunogen or antigen enters into your body, so this is the lag phase. This is the when it enters, it slowly start getting contact with immune cells. Okay, then the log phase, steady rise in the titer of antibody. So whenever they will get contact, those immune cells will get active, and through different mechanisms they will start producing antibody. Okay, so in this log phase your body will start producing antibody. Now what is plateau phase? Because when antibody is getting produced, antibody will then react with antigen and it will neutralize those antigens. So that time equilibrium between antibody synthesis and catabolism. So in equilibrium between antibody synthesis and then neutralization of the antigen. And decline phase, where antibody production will get declined, because your all antigen will be neutralized, you are getting rid of the infection, so antibody production will also get decreased. Now this is a multi-step events. So what happens? Antigen enters, so it comes in contact with antigen presenting cells. So antigen presenting cells, what they do, they present those antigens. They at first sense those antigen, and then they engulf those, and then by antigen processing mechanism they present those fragments, protein fragments from antigens, and present to the different other cells. So some antigen presenting cells are macrophages, dendritic cells. Then processing of antigen happens, like this is the part of antigen presentation. What happens? Presentation of antigen with MHC2. Okay, with the help of MHC2 it presents, and then those MHC2 and presented antigen will react with T cell receptor, so that activation of T cell happens, and then followed by activation of B cell happens. So for B cell it's of two types, plasma cell and memory cells. So plasma cell basically produces antibody, and memory cell is recognize the antigen and it's stored for future infections. Okay, so now we'll discuss about polyclonal versus monoclonal antibodies. Polyclonal antibodies, what are they? Antibodies from many clones. Okay, antibody from many clones are called polyclonal antibodies, and monoclonal antibodies from single clone. So whenever you are getting an antibody from a single clone is monoclonal. So you can generate monoclonal antibody using hybridoma technology. So this is the antigen, okay, and so it has different epitopes, 1, 6, 1, 2, 3, 4, 5, 6. So what is happening? These are different plasma cells, and these are producing antibodies against different epitopes. Okay, so what is that? Then you are getting these antibodies against this antigen from different epitopes, that means from many clones of B cells. So that is polyclonal. But if your plasma cell produces only only one plasma cell is producing antibodies against this antigen, is called monoclonal. So generation of polyclonal antibody. So this is antigen you are giving, then antigen so you have a hapten antigen preparation, you do immunization in animals, so you have the antigen, okay, you prepare that, and then you inject those antigen into animals, so immunization happens. Then you do testing, so immunization happens, so the animal will start producing antibody. Then you test those antibody generation, you take the bleeding, you take the blood and collect the serum, and purification antibody happens. This is polyclonal antibody purification. Monoclonal antibody production. So what happens? Antigen presentation, then emulsification of antigen, you immunize the animals, you test the antibody generation, collect blood, collect serum, test antibody, and after immunization what you do, you isolate B cells from spleen. So you immunize, so from the B cell antibody will be produced, okay. So you directly isolate the B cells from spleen, and this is called culture of myeloma cells. And then what you do, fusion of myeloma and B cells, you so what you will do after immunization, you collect those B cells from spleen, and also you culture myeloma cells. And what is happening, you fuse the myeloma cells with those isolated B cells, and then you screen for fused cells. So whenever those myeloma and B cells will be fused, you screen for those and you harvest. So in a cell culture myeloma, this is the myeloma cell culture line, so you collect those and then you fuse with those spleen cells, and some cells will be fused, okay. And those myeloma basically grows very fast, so when you are fusing with B cell, those B cell fused with myeloma will also grow fast. So then what will happening, this will grow in large culture, and you will those B cells will produce antibody, okay. Monoclonal antibody presentation. So this is the schematic presentation. Now antigen preparation. Purification of antigen. The antigen used to immunize be as pure as possible. Use of pure antigen reduces the generation of cross-reactive antibodies. Two different methods to produce a large quantity of antigen for immunization purpose. So there are two different methods you can use to produce a large quantity of antigen for immunization purpose. One is purification under native condition and another one is purification under denatured condition. So how do you prepare immunogen? Combine 100 microliter of antigen, that is 100 to 150 microgram, with an equal volume of fluids complete adjuvant to increase antigenicity to a final volume of 200 microliter. Mix thoroughly to obtain the emulsion using a syringe or a pipette. The perfect emulsion of the antigen can be tested by dropping a small amount into a beaker containing water. A good emulsion will not spread on water surface. Okay, this is how you prepare the emulsion. Before immunization, take out 1 to 0.5 ml mice blood from the tail vein before the first injection. Incubate the sample at 4 degrees at 30 minutes and allow the blood to clot. Centrifuge the sample at 7000 G for 10 minutes. Collect the serum and store it at minus 20 degrees Celsius and labeled as pre-immune serum. So what happens? Take out. So if you are taking out 5 mice, so these mice strains are Balbisi strain from the cage and sterile them by spraying 70% ethanol. Then you are injecting 200 microliter of antigen mixture per mice. During this step, either use a helper to hold the mice or use a restraint device to hold the mice. Briefly clean the injection site with 70% ethanol and inject antigen through multiple routes. Okay. So those routes can be intravenous. Antigen mixture can be directly injected into the tail vein. Intraperitoneal injections. While making injection, avoid injecting the antigen into the stomach. Okay. Subcutaneous and intramuscular. So you have to know all this process. So you can inject intravenously, you can inject intraperitoneal, you can do subcutaneous and intramuscular injection into the tight muscle. After injection, keep the mice back to their cage, obviously. Then combine 100 microliter of antigen with an equal volume of fluids in complete adjuvant to a final volume of 200 microliter. Mix thoroughly to obtain the emulsion using a syringe. So you have to mix those adjuvant and antigen basically, and after 4 weeks and first injection, what you have to do, inject first booster doses. Okay, so you have to inject the booster dose also, so that those antibodies are getting produced subsequently. So repeat booster injection 4 to 5 times after every 4 weeks to generate a robust immune response and development of memory B cells. Okay. Now your antibody will be getting produced. Now you have to determine the antibody titer. Take out again 100 to 500 microliter of mice blood from tail vein before the first injection. Incubate the sample at 4 degrees for 30 minutes, and this is the same procedure, and this is how you will collect the serum and determine the antibody binding efficiency by indirect ELISA. Okay. Monoclonal antibody production we have taught. So preparation of peritoneal exude cells. So peritoneal exude cells, what are those? Peritoneal exude cells derived from the bovine hypothalamus, okay, as a feeder cells for culturing of hybridoma cells. It has following steps. So this is important to know that peritoneal exude cells, what are those? Those you are getting from bovine hypothalamus as a feeder cells for culturing the hybridoma cells. So it has following steps: sacrifice a non-immunized mice either by cervical dislocation or carbon dioxide asphyxiation. Soak the dead mice in a beaker containing 95% ethanol prior to start the dissection of mice in the laminar hood. Make small cut at the abdominal region and expose the peritoneal cavity. Okay, you in the mice you cut in the peritoneal region and expose the peritoneal cavity. Inject 3 to 5 ml of serum free DMEM, okay, 3 to 5 ml of serum free DMEM into the peritoneal cavity using disposable syringe. Flush the peritoneum and collect the peritoneal exude cells. And plate it to the disposable petri dish with 10 ml serum-free DMN media. So only DMN media you are injecting, and then you are again injecting it out. So you are collecting peritoneum cells. Count the cells and dilute the cells to 4 into 10 to the 5 cells per ml. Allow the cells to incubate for 2 days, and possible contamination needs to be checked before using these cells for hybridoma culture. So preparation of spleen cells, how do you prepare? Sacrifice those non-immunized mice either by cervical dislocation. Cervical dislocation means these are mice, so you just do like cervical dislocation, and you dislocate this cervical by so there is a technique that you have to just stretch the mice out a little bit, so you can do that. Or carbon dioxide like you have to, what you have to do, you have to just give enough carbon dioxide so it doesn't get oxygen, so then asphyxiation will happen, so it will die. So this is the process. So then what you'll do, you soak the dead mice in a beaker again to sterilize those. You cut at the abdominal region and dissect to remove spleen. Now you are cutting in the abdominal region and you are dissecting the spleens. Spleens will be in the abdominal region. Okay, you are dissecting out using forceps, and then you are keeping it in a disposable petri dish. Inject 2 to 5 ml serum-free DMM into the spleen. Again you are injecting DMM into the spleen, and you are this tape will swell the tissue. Then what will do, tease the tissue with the help of forceps and release the cells into the petri dish. Remove the debris and cell clump. Centrifuge at 50 G for 5 to 10 minutes at room temperature. Incubate the cells in a Gray's hemolytic solution. 8 ml of Gray's A solution and 2 ml of Gray's B solution. This tape will remove the RBCs from the spleen cells, leaving myeloma cells. So in this, this tape is important because you are incubating those cells with a Gray's hemolytic solution. What it will do, it will do hemolysis. So if it does hemolysis, what will happen, all the RBC will get degraded and only those myeloma cells will be there. Then you collect the cells and plate in the tea flask and grow up to mid-log phase and resuspend the cells in a serum-free DMM media at room temperature. Fusion of spleen and myeloma cells. Mix spleen and myeloma cells in a ratio of 5:1 or for 10:1 in a sterile centrifuge tube. Centrifuge the cells at 120 G for 5 minutes at room temperature and remove the supernatant. Gently tap the bottom of the tube and add 1 ml of 50% PEG 6000 in serum-free media. PEG solution should be added dropwise to avoid clumping of the cells. Then dilute the mixture by adding 3 ml worm serum-free DMEM over a period of 1 to 2 minutes. Centrifuge the fused cells and resuspend the cells in DMEM containing 20% FCS at a cell density of 10 to the 6 or 10 to the 5 cells per ml. Then add feeder cells in 96 well plate on the top of this and add 50 microliter of fused cells. Incubate the cells at 37 degree 5% CO2 for 24 hours prior to go to next step for screening those hybridomas. Now the selection of hybridoma cells. So hybridoma cells contain spleen cell capable of coding antibody, whereas myeloma cells does not produce antibodies but has ability to grow indefinitely. So if you incubating of hybridoma cells in a hat medium allows growth of fused cells, whereas unfused individual myeloma cells or kidney cells does not be able to grow. So these are B cells which has HGPRT, which is HGPRT plus. So if you are fusing, it with myeloma cells, then fusion will happen and some will be unfused, normal B cell and then normal myeloma cells. So then you are you are incubating it hat medium, so and those will be selected which are fused. Okay, so large scale culture of hybridoma cells, unfused B cells undergo normal cell death. Okay, which are normal cell death, and myeloma cells cannot grow in hat media. So myeloma cells cannot be grown in hat media, so that's why those are getting died, and unfused B cells will undergo normal cell death because these are not infinitely grown. Okay. Now molecular mechanism of hybridoma selection. So what is the molecular mechanism of hybridoma selection? Now we will discuss about this hat medium we were talking about. So we'll discuss about the molecular mechanism of hybridoma selection. So the de novo pathway is, you have simple sugar. So from simple sugar you are getting nucleotides. From that B cells AGPRT plus, hybrid cells, and myeloma cells is AGPRT minus. So de novo pathway is blocked in all cells. And what is salvage pathway? When you have hypoxanthine, so with the help of AGPRT it produces guanine, and then your and this will not happen in the case of myeloma cells. So what is happening? Nucleic acid synthesis is essential. You know that nucleic acid synthesis is essential for growth and multiplication, and if you can hinder any of these methods, it won't allow the cell to grow. So HAT medium inhibits de novo pathway, okay, and cells with salvage pathways. So what is the principle is that when you are growing your cell in HAT media, so HAT media has certain substance like aminopterin and so on, which will not allow this de novo pathway of making nucleotides, okay. But cells have also a salvage pathway, and this salvage pathway, what happens, AGPRT, whichever cell has AGPRT enzyme, it will produce guanine or nucleotides from hypoxanthine. But if one cell doesn't have this AGPRT, that is AGPRT minus cells will not able to access this salvage pathway. So eventually in HAT media, when they won't be able to access de novo pathway, they will die, okay. So HAT media inhibits de novo pathways, and cells with salvage pathways, that is containing AGPRT enzyme, can survive. Myeloma cells depends solely on de novo pathways for nucleic acid synthesis, whereas kidney cells has both de novo and salvage pathway, okay. In the presence of HAT, individual myeloma cells or kidney cells will not survive, whereas only hybridoma cells or hybrid cells can be able to survive. Now after you got the hybridoma cells, you have to select those. Once the colonies are observed, isolate these cells by serial dilution methods. Delineate the boundary of each colony with a marker from the backside of the plate. Remove the media and put cloning ring to each colony. Wash the colony with PBS and add 100 microliter of trypsin EDTA to remove the colony. Wash the colony with PBS and transfer into 1 well of 24 well plate. So you are basically doing antibiotic selection, and then you are diluting those colonies. So you are delineating the boundary of each colony with a marker from the back side of the plate. So you are just selecting those colonies individually, and remove the media and put cloning ring to each colony. Wash the colony from PBS and add 100 microliter of trypsin EDTA to remove the colony, and wash the colony with PBS, okay. Allow it to grow and become 80% confluent. Then you transfer the cells to a 6 well dish in the presence of selection media again, and allow it to reach 80% confluency. Take a small aliquot of the cell and test the presence of antibody with ELISA. This is how you select your hybridoma cells or fuse cells. Now we'll be going to the part 2 of immunology. So this is antibody antigen cognate pairs. So this is the antigen, and it has different epitopes where it has different sites where those antibodies are getting bound. So these are the different epitopes, and this is antibody. So what happens, antigen antibody interaction can be because of salt breach, okay. It can be hydrogen bonding, it can be van der Waals, it can be pi-pi interaction. So this antibody can be insoluble or soluble. If it is insoluble, it shows the property of agglutination. It can be soluble also, it can get precipitated, radial diffusion assay. So you can get to know by precipitation, radial diffusion assay, and immunoprecipitation. And some different immunoassay will also discussed. Will also discuss. Those are ELISA, Western blot, and radioimmunoassay. So what are agglutination reactions? Agglutination is the process of linking together of antigens by antibodies and formation of visible aggregates. So agglutination is a process by which you are providing antigen and antibody. Those are getting cross-linked with each other and forming a visible aggregate. Agglutination reaction involve particulate antigens such as bacteria, virus, or RBC. That means those are not soluble. So by agglutination you can prove those, and very sensitive and reproducible. There are two types of agglutination reaction: direct agglutination reaction and indirect. So what is direct? Direct agglutination test diagnoses antibodies against a large number of cellular antigens such as RBC, bacteria, and fungi. This test is carried out in plastic microtiter plates that have several small shallow wells. The formation of antibody-antigen mat which sinks to the bottom of well. However, in the negative reaction, agglutination does not occur and insufficient antibodies are present to cause the linking of antigen. So this is antigen-antibody combination. So what is happening? Agglutination is happening when antibody are cross-linked, like those. So what is happening? Basically, antibodies are interacting with antigen, and then if we are applying anti-immunoglobulin also, that means immunoglobulin means immunoglobulin fold, antibody has immunoglobulin fold, so anti-immunoglobulin will cross-link different antibodies. So then this agglutination is happening. Okay. So here you can see agglutination happen, and this is negative little bit amount. So clumping of antigen will make it. So in the case of RBC, if it's getting like clumping of antigen, so basically it is showing red because of the for your visualization, that if clumping happens then you will see a red dot, and no clumping you won't be able to see. So you can also do blood group typing by this method. So blood typing is accomplished by mixing drops of blood with sera, that means antibodies, against ABO and the RA system. If the blood cells clump or agglutinate, the antibody has bound to the appropriate antigen on the cells. Your blood type matches whatever antibody caused agglutination. That is, if your blood agglutinates in anti-B, you have the B antigen under type B. So blood types are indicated by both the ABO and RH antigens present. So that means if your blood group is A, then if you are giving anti-A, that means antibody against A, so agglutination will be happening. But if you are giving anti-B, agglutination won't be happening. For blood group B, anti-B will produce agglutination. For AB, both those antibody will give agglutination. But for A, none of those will give agglutination. Okay. Now those are the direct agglutination. Now we'll discuss indirect agglutination methods. Adaptation of soluble antigen for agglutination reaction. This type of diagnostic tests are very rapid, particularly for the detection of soluble antigens such as streptococci. In the antigens, if the antigens are absorbed onto particles, that means example RBCs, latex beads, soluble antigens can respond to agglutination test. Antibody reacts with the soluble antigen adhering to the particles. Therefore, the particles agglutinate with each other as these do in the direct agglutination test. So one example is hemagglutination. Hemagglutination is a phenomenon of clumping of RBCs when the RBCs are agglutinated by certain viruses such as those causing mumps, measles, influenza, etc. So this is RBCs suspended in PBS. RBCs settle down to the bottom of the well, and view from top is this. But if RBCs is suspended in PBS and you are adding virus, so what will happen? RBC won't be able to settle down because viruses are adjoining those antibodies, and RBC agglutination is happening. So and if you view from top, then RBC button you won't be able to see. So hemagglutination is a linking of our red blood cells by the virus particles in an isotonic solution that result in clumping. The clumped RBCs settle down much slowly as compared to virus-free RBCs. Thus, the untreated RBCs settle down to the bottom of the well and forms a defined red button, which is not observed in the case of virus-mediated cross-linked RBCs. So if you want to perform this experiment, what you need, you can have RBCs from any organism, V bottom microtiter plates. You have to have this kind of microtiter, V bottom microtiter plate, 1x PBS, micro tips and pipettes, conical tube, centrifuge with swinging bucket rotor and bleach solution. So now you have to prepare chicken RBCs. You can also have mouse RBC. So you have to collect 1 to 2 ml of chicken whole blood with so as you are so what we'll discuss why you are having these chicken RBCs. So 1 to 2 ml of chicken whole blood with EDTA. EDTA means it's anticoagulant, so your antibody like RBCs won't get coagulated. Fresh RBCs should be used for the assay. You spin at 1500 rpm for 10 minutes. Carefully remove the white buffy layer on the top of the RBC pellet. Remove the serum. Dilute with 3 ml 1x PBS and mix by inverting. Spin at 1500. Discuss the supernet and repeat. Make a 1% chicken RBC solution using 1x PBS and dilute. Now viral dilution assay. You have to, so as you are infecting those RBCs with virus titers, so they have affinity to coagulate, so that's why we are using it. So to the first well, what you'll have, you add 50 microliter of virus sample and mix thoroughly, and transfer 50 microliter of the first well to the second well, and that is how we will do dilution. Then discard 50 microliter from the last well into a bleach solution. Okay, so why bleach? Because bleach will decontaminate the sample. Okay, then prepare appropriate a positive and negative control. The positive control should be from a sample where the virus is already known to be present. The negative control where only RBC and PBS are present, no virus. Add 50 microliter of the 1% chicken RBC to all the wells. Working concentration is 0.5%. Mix gently and close the lid. Keep in room temperature 15 to 30 minutes to develop the clumping behavior, and you will get to see this. These are positive controls, this sample 1, sample 2, and these are negative controls where there is no virus. So now you will be able to calculate the titer through this calculation. Okay. Now as we are discussing antibody-antigen interaction, we will now discuss precipitation reactions. The reaction of soluble antigens with IgG or IgM antibodies to form a large interlocking aggregates is called precipitation reaction. The precipitates formed by antibodies are known as precipitins. It occurs in two states: rapid interaction within a second between antigen and antibodies and formation of complex, slow rate of reaction completing even within a few minutes or hours and forming lattices from antigen-antibody complexes. Mismatch of antibodies and antigen ratios, no visible precipitate is formed. Immunodiffusion test. So immunodiffusion tests are performed in a gelled agar media. One of the immunodiffusion test is Oucher colony test. In Oucher colony test wells are cut into which purified antiserum, a serum containing antibodies, is added, and to each surrounding well soluble form of test antigens are added. Therefore, a line of visible precipitate is formed between the wells where after diffusion optimal ratio of antigen antibody is formed. Okay, so you are adding antigen 1, antigen 2 and antibody. When they are diffuse and come together, you will have a precipitation line. Radial immunodiffusion. Radial immunodiffusion is a quantitative immunodiffusion technique used to detect the concentration of antigen by measuring the diameter of the precipitin ring formed by the interaction of antigen and antibody at optimal concentration. So in this method, the antibody is incorporated into the agarose gel, okay, and whereas the antigen diffuses into a radial pattern. Thus the antibody is uniformly distributed throughout the gel. So this is agarose gel which is in having the antibody. The diameter of the precipitin ring is proportional to the concentration of antigen. With increasing concentration of antigen, precipitate rings with larger diameter are formed. The size of the precipitate rings depend on antigen concentration of the sample well, antibody concentration in the agarose gel, size of the sample well, volume of the sample. So the diameter of the precipitin ring is proportional to the concentration of the antigen. Antigen concentration, the diameter of the ring increases. So with increasing concentration of antigen, precipitate rings with larger diameter are formed, and the size of the precipitate ring depends on what? It depends on antigen concentration in the sample well, it depends on the antibody concentration, size of the sample well, and volume of the sample. The standard curve can be obtained from which one can determine the amount of an antigen in an unknown sample. This is the diameter of the precipitin ring, and this is antigen concentration. If more than one ring appears in the test, more than one antigen antibody reaction may have occurred. This could due to a mixture of antigens and antibodies. This test is commonly used in the clinical laboratory for the determination of immunoglobulin levels in patient samples. Materials: glassware, you have conical flask, measuring cylinder, reagents, agarose, assay buffers, standard antigens, test antigens, glass plates, gel puncture, distilled water. Equipments: you have incubator, microwave, Bunsen burner. So procedure: so you have to prepare 10 ml of 1% agarose gel. Take 6 ml of this gel solution in a clean test tube. Allow the solution to cool down to 55 to 60 degrees Celsius and add 80 microliter of antiserum to 6 ml of agarose solution. Mix well for uniform distribution of the antibody. Pour agarose solution containing the antiserum onto a grease-free glass plate placed on a horizontal surface. Punch wells with the help of gel puncture corresponding to the marking on the template. Use gentle suction to avoid forming rug wells. Add 10 microliter of given standard antigen and test antigen samples to the well. Incubate the glass plate in a moist chamber overnight. Then what will happen? Observe the precipitant ring surrounding the antigen well. These are antigen well, and mark the edges of the precipitant ring and measure the diameter. You have to measure the diameter. So some troubleshooting surrounding these experiments are: if you face problem in getting no precipitant ring, so the probable cause are inadequate filling of the wells, drying of the agarose gel during incubation, inactivation of antiserum. Sample should be loaded directly into the well without spilling of the sides. Ensure that moist chamber has enough moist cotton to avoid drying of the gel. Antiserum should be added to the agarose gel only after the temperature reaches 55 to 60 degree. Blue precipitant ring observed. If you observe blue precipitant ring, inactivation of antiserum, uneven pouring of gel. Antiserum should be added to the agarose gel only after the temperature reaches 55 to 60 degree. Place the glass plate on a flat surface while pouring the gel. Do not move the plate once the gel is poured. Now we will discuss immunoelectrophoresis. The principle of immunoelectrophoresis is based on the movement of antigen and antibodies to opposite poles after applying electric field. If a reaction occurs, the precipitated line you will observe. It is useful for the diagnosis of bacterial meningitis and other diseases. This is antibodies moving at this direction and antibodies moving at this direction, and you will have this precipitation line when antigen will bind to antibody. Now we will discuss how to harvest monoclonal antibodies. So what happens once the color of the media changes from red to yellow orange? Change the media with a fresh DMEM. The healthy cell lines that produces antibodies transfer to 24 well dishes containing 30 to 60 ml of complete media for the large production of antibody. Harvest the supernatant by transferring culture into a tube and centrifuge it 120 G for 10 minutes. Transfer the supernatant to fresh tube and adjust the pH and add sodium azide. Antibody purification. Antibody purification using antigen coupled column. So this is affinity column, this is samples, washing state, receptor ligand interaction, then elution. Okay. So this is very much similar to as affinity purification, because you are also purifying with affinity column. So we have discussed affinity purification. Now the same process is that you are binding antigen with like antibody, and then your sample has affinity with the affinity column. You are washing so that all the undesired items will get washed out, and then you will elute with strong ligand, so that uh, you are purifying your protein of interest or sample of interest. Now we'll discuss immunoprecipitation. So for immunoprecipitation, again the concepts are similar, that you are precipitating antigens with the help of antigen antibody interaction principle. So you have bits and your antibodies are attached within the bits, and and you have also cell, you are collecting cell lysers. So some particular antigen will bind to antibody, and then you will identify those antibodies because those are the only antigen will bound to antibody specifically. So for materials, you have tris HCl buffer, NaCl, NP40 detergent, PMSF is a protease inhibitor. For lysis buffer, you will have all this. Washing buffer, you have again a tris HCl buffer, EDTA, NaCl, and NP40 detergent. So you have to prepare cell lysate. Wash approximately 1 million cells with a phosphate buffer saline and add 1 ml of ice cold lysis buffer in the cell pellet. Similarly, approximately 50 mg tissue lysate can also be prepared. Centrifuge the tube at the maximum speed for 15 minutes at 4 degrees Celsius while shaking gently. Step 2. Incubation with antibody. To the supernatant, keep adding a specific antibody of appropriate concentration for 1 hour at 4 degrees Celsius while shaking gently. Add 0.9 ml of the protein A cephros slurry and shake for another 30 minutes at 4 degrees Celsius. Wash the A cephros mixture thrice in washing buffer containing 900 millimolar NaCl to remove the non-specific binding. Finally, wash the mixture once in a washing buffer. Procedure: analysis of sample on SDS page. Remove the supernatant after centrifugation at high speed. To the protein A cephros slurry bits, add 800 microliter of 1x SDS gel loading buffer, and then you make the sample for SDS page. Visualize the protein bands by staining chromacy blue or silver stain. So here you have loaded the marker, only antibodies, 2 beads plus lysates, and 3 is antibody plus beads plus lysates. The interacting proteins will give an extra bands in SDS page and can be further characterized by probing the proteins on SDS page with other antibodies in Western blots. Now, from the extra bands, you will be able to understand which are your lysates, and you can do Western blot for specifically identify your protein. Now you can also do pull down assay. So for pull down assay, you have a GST added with, so you have a GST and the cell, you will get some cell lysate. So then you are basically, if your protein has affinity with binding with the affinity column, then you are basically pulling out. So in the what you will do, you will wash and identify the protein, and with the GST you will have a probe attached, and if your probe is binding with the bed, and then you are basically purifying the bed with the probe. So we will discuss in detail. So again you will have those lysis buffer common. Just before use, add 1 millimolar of PMSF as protease inhibitor. GST fusion protein carrying the bed, then bed which is GST protein, 2x SDS sample buffer you need, glutathione agarose beads, boiling water bath, and microfuge. So pre-clean the lysate. Initially pre-clean the lysate by incubating approximately 1 ml 50 milligram tissue, and then you add 50 microliter of 50% slurry of glutathione agarose beads in a lysis buffer, and you centrifuge it. Setting up the reaction, divide the clean lysate into 2 tubes equally and add 50 microliter of glutathione agarose beads. To one tube, add approximately 10 microgram of GST protein, and to the other tube add approximately 10 microgram of GST fusion protein. The amount of probe GST fusion protein and the control protein added should be equimolar in the two reactions. Incubate the tubes for 1 hour at 4 degrees Celsius with mixing. Centrifuge the samples at the maximum speed for 5 minutes in a microcentrifuge. Save the supernatant at 4 degrees Celsius in a fresh microfused tubes for SDS page analysis. Wash the beads with 1 ml of ice cold GST lysis buffer twice using centrifugation at top speed for 1 minute. Then you will do elution. You elute the GST fusion protein and any proteins bound to it by adding reduced glutathione in a tris buffer. Preparation of the sample for SDS. So these are very much similar as affinity purification we have covered. You can do Kumasi blue, you can do immunoblot to shows the possible protein interaction. This is the marker. One is GST and probe, second is GST, and third is only probe. Now we'll discuss ELISA. So ELISA is an immunological technique used to measure the level of antibodies or antigen in the body fluid. It has been used to diagnostic to identify the antigen or cross-reactive antibody. ELISA can be performed in different ways to either measure antibody or antigen. So you are doing direct ELISA where antigens are attached and then you are adding conjugating the primary antibody, and with substrate you are getting light up. And also indirect ELISA where secondary antibody conjugation will happening with primary antibody and you are getting the light. And sandwich ELISA where you have the capturing antibody attached with the bit and you are adding antigen, and against this antigen you are adding another primary antibody and secondary antibody, and this is sandwich ELISA. Okay. And competitive ELISA where you are also you can add inhibitor antigen so that those antibodies will get bind with the antigen, and so this is basically competitive interaction between two antigens to getting bound with the antibody. Now direct ELISA. So what happens, the target antigen is first coated into the bit, the multi-well plate, then you are incubating with primary antibody and you are adding some specific substrate so that this gets light up. So this is very simple and quick to perform this experiment. Disadvantage, specificity of the primary antibody may be affected by the enzyme linking. Linking primary antibody to for each specific ELISA experiment is expensive and minimal signal amplification. Indirect ELISA, this setup is used to measure the level of antibodies in the serum, used to calculate the titer of the antibodies, and in indirect ELISA setup a known amount of antigen is coated to the well and it's incubated with the different dilutions of antibodies. So the antigen bound antibody, is again recognized by a secondary antibody. So the principle is like Western blot. Okay, if you have any query, please let me know. So for that, what are the materials and reagents required for this ELISA performance? You prepare a 50 millimolar bicarbonate buffer pH 9.2 at distilled water and filtered, sterile. ELISA plate, flat bottom 96 well plate is more suitable to perform ELISA. Antigen solution, prepare 5 microgram per ml antigen solution. You prepare BSA, primary antibody, secondary antibody dilution, and PBS. Procedure, protein coated container. You have antibody bind to protein, excess washed away, visual changes confirms the correct protein. So you are before that you have to add secondary antibody and then you are adding substrate, so visual changes happens. So for coating, what you have to do, prepare 5 microgram ml of antigen solution in a bicarbonate buffer and dispense 50 microliter per buffer and put it overnight, so you are getting a coating. Block each well with 1% BSA in bicarbonate buffer for overnight. Then you are doing different dilutions and you are adding antibody and 1% BSA, so total volume is this, and dispense 50 microliter of each dilution in respective wells and incubate for 45 minutes at 37 degrees. You do wash 4 to 5 times with PBS plus 1% to 20. Secondary antibody, prepare appropriate dilution of secondary antibody and then disperse in 50 microliter per well. Wash 4 to 5 times with PBS plus 1% to 20. Development, dispense 1 mg per ml OPD plus H2O2 in citrate buffer. Stop the reaction by 7.5% H2S4 and take absorbance at 460 nanometer. So this is the results. So this is absorbance you get at 460 nanometer. These are different wells of 96 well plate. Antibody dilutions where you have more absorbance, you can see from the curve. Now we'll discuss sandwich ELISA. So sandwich ELISA, this setup is used to measure the level of antigen again. So this, you know, that sandwich ELISA means you are capturing one antigen with two antibodies, and you are getting colorimetric you are using colorimetric substance, so that binds with secondary antibody and then it's getting light up. So in this method, a capture antibody is used to collect the antigen from the sample. Afterwards, secondary antibody is used to detect antigen bound to the capture antibody. Second antibody is directed against the antigen using a unique distinct epitope. The antibody is linked to the biotin and that can be recognized by the avidin streptavidin HRP complex. In the last step, peroxidase substrate is used to get a readout. Material and instrument. So we have to capture antibody, detection antibody, enzyme, reagent, standards, enzyme reagents, streptavidin HRP conjugate. So anti-mouse TNF monoclonal antibody is supplied as one vial. Biotin anti-mouse TNF monoclonal antibody is supplied as one vial and I recommended 1:500 dilution. Enzyme reagent, streptavidin horse redox peroxidase conjugate, this is a reagent. Recombinant mouse TNF is supplied as lyophilized powder, so this is the standards, and enzyme reagent streptavidin HRP conjugate. Coating buffer, you have sodium phosphate buffer as a coating. Acid diluent, PBS you can use with 10% APS as acid diluent. Wash buffer, you have PBS with 220. Substrate solution, then stop solutions, micro plate reader, micropipettes, these are the required items. Specimen collection and handling. Specimens should be clear. So specimens should be clear, non-hemolyzed and non-lipic. In the case of cell culture, remove any particular matter by centrifugation and assay immediately or store samples at less than -20 degrees Celsius. Avoid repeated freeze-thaw cycles, whereas in the case of patient blood, use a serum separator tube and allow samples to clot for 30 minutes. Preparation of TNF alpha standard dilution. So dissolve the vial content into 1 ml deionized water to yield a stock standard 30 nanogram per ml. So this is TNF alpha concentration, and you are doing dilution using a dilution buffer. So this is from stock solution, you are diluting it at like serially. So ELISA plate coating, you have to coat the plate, you have to block with BSA. Then what will do, you detection. So now you will be add 100 microliter of working detection antibody plus HRP to each well and incubate for 1 hour. Aspirate the detector solution and wash 5 to 6 times and add 100 microliter of sustained solution to each well and incubate for 30 minutes at dark. Stop the reaction by adding 50 microliter of stop solution to each well and read the ELISA plate at 450 nanometer and at 570 nanometer if it's required to substrate background absorption. Okay. So this is the result determination of TNF alpha level from this plot. And what is competitive ELISA? Competitive ELISA is based on the competition binding for the primary antibody between the target antigen in a sample and the same antigen that is coated to the multi-well plate. The primary antibody is first added to the sample to form antigen-antibody complexes. The sample is then added to the wells that are coated with the target antigen. Okay. Only unbound primary antibody in the sample can bind to antigen coated in the wells. Hence, the more antigen in the sample, the less uh, so the more antigen in the sample, the less antibody is available to bind to antigen in the wells. So advantages, this is high sensitivity, crude samples can be used, signal can be quantified by comparing in a serial dilution standard curve, and disadvantages, requires primary antibodies with high specificity to the antigen. So, and also we will discuss Western blot. So Western blot also we already discussed. The concept is very much similar. You run a gel, and then in the gel your protein will get separated. You add primary antibody, and you will wash excess, and you will again add secondary antibody, again wash, and then add substrate and visualize. So the materials, you can get proteins from E. coli cells or mammalian cells. Then you have to have protein standard markers, transfer buffer, transfer membrane, nitrocellulose membrane or PVDM membrane, plastic trays, spatula, blotting sheets, semi-dry electroblotting unit, reagents for performing electrophoresis, anti-GFP antibodies, why anti-GFP? Because you were having GFP protein, and anti-rabbit and IgG, HRP, and developing reagents. So at first you have to prepare your sample. The sample preparation is very much similar with others, and the main thing is that the transfer of protein gel on blotting membrane. So you have to prepare the transfer membrane. Cut the membrane of the sample size as gel. For nitrocellulose membrane, place the membrane in the transfer buffer and observe that the liquid has wicked the membrane. Areas appeared as white spots need special consideration. For PVDF membrane, immerse the membrane into the 100% methanol for 50 to 30 minutes. Decant the methanol and submerge the membrane into the transfer buffer for additional 10 to 30 minutes. Assembly of transfer cassette. So the transfer cassette assembly also we discussed. So what we have to have, you have the filter paper, nitrocellulose membrane, gel, filter paper again, all will be soaked in the transfer buffer, and then you have cathode and anode, and you will add to the electrophoresis chamber. And then what will happen, from the gel the protein will go to the blot. So after your transferring is done, you have to wash the membrane with distilled water to remove any remaining poncho stain. So basically, if you can use after this transferring, you can detect your protein by poncho stain, but you can avoid this step also. You can directly go for blocking, and followed by primary antibody incubation. Then probing. In Western blotting, probing can be done in two ways. Two-step probing. In two-step probing scheme, membrane is first probed to a primary antibody to recognize the protein of interest. Generation of monoclonal or polyclonal antibody is discussed already. Membrane is probed with the primary antibody with an appropriate dilution for 1 hour. Membrane is washed with buffer containing non-ionic detergent and reprobed with another antibody detected against the primary antibody. The secondary antibody is then coupled to its enzyme, and you add a substrate and get the detection done. And one-step probing. In one-step probing, primary antibody contains enzyme or fluorescent level. Okay, and then one-step probing is not very common. Blot development. There are multiple ways to develop the blot and detect the protein of interest. Two different reagents are available to develop Western blots. So HRB based reagents, example fluorochloronaphthol, DAB, TMB. And what happens? Oxidized product to form purple precipitate. DABs in NiCl2, what happens? Forms dark brown precipitate. TMB forms dark purple stain. Alkaline phosphatase, BCIP, NPT. BCIP hydrolysis produces indigo precipitate after oxidation with NBI. Luminescent HIP based luminol H2O2. Oxidized luminol gives off blue light. Alkaline phosphatase substituted 1,2-dioxetane phosphatase. Dephosphorylate substrate gives off light. So colorimetry, it can be colorimetric detection, or it can be chemiluminescent detection. So wash the membrane with TBS to remove detergent. Place a membrane into the colorimetric reagent and protein bands appeared in 10 to 30 minutes. Stop the reaction by washing in distilled water. Air dye the membrane and photograph for permanent record. Chemiluminescent detection. The different chemiluminescent reagents are given in the table. Transfer the membrane into chemiluminescent reagent. Soak the membrane for 30 seconds to 5 minutes. Drain off the reagent and wrap the membrane into the plastic wrap. Place it in film cassette and expose the membrane to film for a few seconds to several hours. Fluorescent detection. Secondary antibodies leveled with fluorescent dye and captured in the scanner. So this is how the Western blot happens. You are binding antibody, primary antibody, then secondary antibody, and then your photographic film shows bands of desired protein. If you have any doubt, feel free to ask. So today also we'll be discussing some examples, questions of related to immunology from previous year, so that it will help you in solving this year's assignment. So only one part is left for today's discussion. It's cellular response. Okay. So it has some receptor, you can see TCR, and some adapter proteins are getting bound. Those are, so basically those are part of cellular signaling or cell signaling. Those adapter protein will induce other proteins, and then it induces the gene expression through NF kappa beta. So this is gene expression, metabolism. Some proteins are associated, cytokines. So what are the principles or experiments you can do to recognize microarray, NMR, proteomics, and so on. So what are cytokines? Cytokines and chemokines are extracellular signaling molecules that mediate cell-cell communication. Okay, they are released from the cells and have critical roles in many biological processes such as cellular growth, differentiation, gene expression, migration, immunity, and inflammation. In most biological processes, multiple cytokines operate in a large network where the action of one cytokine is regulated by the presence or absence of other cytokine. So how we can measure so many cytokines from cell? ELISA. ELISA is an immunological technique used to measure the level of antibodies or antigen in the body fluid. It has been used to to identify the antigen or cross-reactive antibody. ELISA can be performed in different ways to either measure antigen or antibody. So we have already discussed different kinds of ELISA. Cytokine array. So this is cytokine array chip, and these are the samples. You are incubating the sample, and you are incubation with biotin-related antibody. So what happens? You are incubated with labeled streptavidin, that those will bind to the biotin-related antibody, and you will get data. So what is cytokine array assay? Carefully selected capture antibodies have been spotted in duplicate on nitrocellulose membrane. The sample or antibody mixture is then incubated with the mouse cytokine array membrane. Any cytokine detection antibody complex present is bound by its cognate immobilized capture antibody on the membrane. Following a wash to remove unbound material, streptavidin, HRP, and chemiluminescent detection reagents are added sequentially. Light is produced at each spot in proportion to the amount of cytokine bounds. So this is the principle is very much similar to the ELISA. So the same process, you have to make the sample, serum, then you have to isolate cytokines. So reagent preparation, mouse cytokine array, 4 nitrocellulose membrane each containing 40 different anti-cytokine antibodies printed in duplicate. Handle membrane only with glove so that you are not adding any protein contamination. Detection antibody cocktail, 1 vial of lyophilized biotin-related antibodies. Before use, reconstitute the detection antibody cocktail with 100 micrograms deionized or distilled water, 1:1 buffer, chemi reagent mixture you have to use. Assay procedure. So bring all reagents to room temperature before use. Keep samples on ice to avoid contamination. Wear gloves while performing the procedure. So you have to prepare all the reagents. So you have to prepare all the reagents and samples as directed in the previous section, and pipette 2 ml of array buffer 6 into each well of the 4 well multi-dish to be used. Using flat tip tweezer, remove each membrane to be used from between the protective sheets and place in the well. Incubate 1 hour with rocking platform shaker. While the membrane are blocking, prepare samples by adding up to 1 ml of each sample to 0.5 ml of array buffer. Add 15 microliter of reconstituted mouse cytokine array panel detection antibody cocktail to each prepared sample. As a procedure, aspirate array buffer 6 from the wells of the 4 well multi-dish and add sample antibody mixtures. Incubate overnight at 2 to 8 degrees Celsius on a rocking platform. Carefully remove each membrane and place into individual plastic containers with 20 ml of 1x wash buffer. Wash each membrane with 1x wash buffer for 10 minutes on a rocking platform shaker. Repeat 2 times for a total of 3 washes. Dilute the streptomycin HRP in array buffer and I will conjugate. Carefully remove each membrane from its wash container. Allow excess buffer to drain from the membrane. Return the membrane to the 4 well multi-dish containing the diluted streptomycin HRP, and then you incubate at room temperature. So after removing each membrane from the wash, allow excess wash buffer, and then you will add chemi reagent and you image those samples in the chemidome. So this is untreated, this is treated. So these are some reference spots. So where your protein of interest is, those primary antibody and secondary antibody will bind accordingly and give you color. So how to analyze data? The positive signal seen on developed film can be quickly identified by placing the transparency overlay template on the array image and aligning with the pairs of reference spots in 3 corners for each array. Note, reference spots are included to align the transparency overlay template and to demonstrate that the array has been incubated with streptomycin HRP cloning during the assay procedure. Pixel densities on developed X-ray film can be collected and analyzed using a transmission mode scanner. Create a template to analyze pixel density. Export signal values to a spreadsheet file for manipulation in a program. Determine the average signal of the pair of duplicate spots. Subtract an average background signal from each spot. Compare corresponding signals on different arrays to determine the relative change in cytokine levels between samples. So this is how we'll do the analysis. This is untreated, this is treated. You overlay this and then you see the extra protein spots you are getting. Now we will discuss the previous year assignment questions so that it will help you in understanding. So first question is, which of the given immune cells are most effective in destroying intracellular pathogen? So is it killer T cell, plasma cells, T helper cell, B lymphocytes? So which of the given immune cells are most effective in destroying intracellular pathogen? So it will be killer T cell. This is the most effective. In ELISA, the reaction between enzyme and substrate before taking the absorbance can be stopped by is treating by H2SO4. Okay, you have just read that the reaction you can stop by adding sulfuric acid or H2SO4. In sandwich ELISA, the capture antibody, what it does, it recognizes iodine streptomycin HRP complex, acts as a secondary antibody, binds to primary antibody, binds to antigen in sample. It binds to antigen in sample. So one antibody will bind to sample, and then capture antibody will also bind to sample. Okay. 2-in-20 is used in Western blotting to reduce non-specific interaction, to reduce non-specific binding and protein-protein interaction, because it's a detergent, it's a mild detergent. A reduction in signal of competitive ELISA implies lack of specificity, more antibody retained in the wells, more antigen in sample, more antibody in sample. So if you are getting reduction in signal in competitive ELISA, that means you have more antigen in sample. In a competitive ELISA, which of the following is true? The secondary antibody must have low affinity for the antigen. The primary antibody must have high affinity for the the primary antibody must have very high affinity for the antigen. So this is true. The reagent used to develop HRP based secondary antibody system in Western blot is luminol hydrogen peroxide. Which of the following is antigen present in cell? B cell, plasma cell, killer T cell, macrophage. Macrophage will be the answer. T cell activation requires T cell receptor to interact with both MHC and MHC1 and MHC2. Antibodies are produced by B cells, plasma B cells. The antigen-antibody interaction involves non-covalent interaction between, not covalent interaction, the interactions are non-covalent between antigenic epitope and Fab region. Okay, so non-covalent interaction between epitope and Fab region. This is the answer. The parameter to define binding capacity of antibody is avidity. Agglutination assay are used in detection of soluble molecular antigens, determining antibody concentration, ABO blood typing, bacterial meningitis. Agglutination assay are using ABO blood typing. Which of the following methods is used to enrich specific protein from the heterogeneous cell? Immunoprecipitation. You can do immunoprecipitation and enrich specific proteins. A researcher is keen in studying protein-protein interaction of a transcription factor X. Which of the following method can be applied? You can do pull-down assay so that your proteins which are interacting with each other will get pulled out together. So these are the few questions we had from previous year assignments. Whenever you are solving this year assignment, it will help you in understanding. If you have any doubt, feel free to get back to me, and we'll discuss in the class. If you have any doubt regarding assignments or tutorial or class lectures. So this is all for today's lecture. We'll again meet next week, Tuesday, 6 to 8 p.m. with week 7 content with the assignments which you had in week 6. You have already solved. If you have any doubt, please let us know. I'll be solving those assignments next day, and also I'll be solving some assignments of week 8 from previous year, and week 7 assignments, and we'll discuss the content of week 8. So thank you for joining. Thanks everyone. We'll stop today's class. Bye bye. Thanks a lot Shifa. Okay, bye bye everyone. We'll meet in next class again with the discussion.